All right, welcome back, everyone. I'm Ben Neal. I'm here to chair the last scientific session before our plenary speaker, Robert Plenge, takes the stage. And I think we're going to move slightly more translationally oriented at the end of the V2F Symposium. And I think that's nice to go from variants to, functions and mechanisms with the idea of getting to therapeutics. So I'll invite our first speaker up here, Vijay Sankaran, a colleague of mine at Boston Children's, Howard Hughes Investigator, and really demonstrates, I think, how basic science can have such clear impact on drug development when you understand the biology. So teach us some biology, Vijay.
Awesome. Well, thank you so much, Ben. And huge thanks to the organizers for the opportunity to come and participate in this amazing symposium. Now, before I get started, I just want to give a little bit of my own perspective because I've been here all day and I've had I've been fortunate to hear a variety of the talks. And I think we started off with Ruth telling us that there are now 9,000 associations with BMI. We know in blood cell phenotypes or height, we have thousands, if not tens of thousands of associations. So in many ways, we should be motivated to try to develop these scalable approaches that you've heard about throughout the day. And certainly we're excited and interested in trying to do that. But my goal in the next 25 minutes or so is going to be to give you a perspective of maybe there's a complementary benefit to really trying to wrestle a single association down to the ground. And so I'm going to try to tell you about one locus and one association that we really want to try to mechanistically and hopefully therapeutically try to really understand. But before I get there, I want to contextualize a little bit of why we do what we do. So each of us is thought to have approximately 100,000 blood or hematopoietic stem cells that reside within our bone marrow and that are responsible for maintaining and replenishing our blood and immune systems throughout our lifetime. Each of these stem cells needs to be able to produce millions of platelets, red blood cells, and a variety of white blood cells throughout our lifetime in order to maintain our health and immune function. Now, we understand a great deal about how this process of blood and immune cell production occurs, and yet most of our understanding of this comes from really elegant and informative studies in model systems, including mice and zebrafish, which have and continue to teach us a great deal about this process. And yet often, when we make observations in our patients or from inter-individual variation in humans, we have challenges in trying to map that directly onto our understanding of this process of blood cell production or hematopoiesis. And so we view this not as a limitation, but rather as an opportunity to take advantage of observations that we make in humans to better understand this process. And our interest in this really started now about 22 or 23 years ago. And at the time, we were interested in trying to understand why the most common monogenic disorders in the world, including sickle cell disease as depicted in this electron micrograph, as well as beta thalassemia, had such incredible clinical variability. We knew that patients with this diagnosis could actually be completely asymptomatic or largely asymptomatic, and yet we knew that many patients could often be severely affected, requiring weekly or more frequent hospitalizations. And at the time, we knew from decades of work that one key factor mediating inter-individual variability in the clinical severity of the disease was the level of fetal hemoglobin that these patients expressed. So typically, during human development, there's predominant expression in utero of this fetal form of hemoglobin, or gamma globin, as shown in the green. And shortly after birth, this switches off, and there's upregulation of the adult beta hemoglobin molecule, as shown in the purple. And this is what is mutated in both sickle cell disease as well as in beta thalassemia. And so continued persistent production of this gamma globin molecule can ameliorate the severity of these beta hemoglobin disorders. And that had been known for decades, yet the precise molecular regulation of this process was not well understood. And so after years of failing to try to understand this process through a variety of systems, we were fortunate because we were able to be involved with some of the earliest genome-wide association studies, initially in non-anemic individuals, and then working with Joel Hirschhorn, we were able to replicate these associations initially in individuals with sickle cell disease. And these studies collectively reveal 3 major regions of the genome associated with inter-individual variation in fetal hemoglobin, including this association on chromosome 2. And these initial observations really led us to go on to functionally show that BCL11A serves as a key and direct repressor of the gamma globin gene, such that when it's well expressed, which is happening in all of your bone marrow cells right now, there's repression of gamma globin and predominant expression of the adult form of hemoglobin. And at the earlier stages of development, there's lower BCL11A expressed at the protein level, and this then de-represses the gamma globin gene and favors production of fetal hemoglobin. But as Melina mentioned earlier today, this of course did not just teach us about a fundamental developmental process, but thanks to an extensive amount of subsequent translational and clinical work, this has now led to the development of now FDA-approved therapies that target this axis, particularly exa-cel or Casgevy, which is from Vertex and CRISPR Therapeutics. And this drug acts by actually taking this repressor of the gamma globin genes, BCL11A, and targeting an erythroid-specific enhancer of this gene to thereby downregulate BCL11A, as Jesse was mentioning, and this thereby de-represses the fetal hemoglobin genes, allowing their robust expression and thereby ameliorating clinical symptoms in patients, as has been borne out. And so this early success really, for us, has served as a motivation to not only think about diseases like sickle cell disease that affect the red cell lineage, but more broadly think about how we could take advantage of human genetic variation across the allelic spectrum to better understand this process. Now, much of our focus has really been on how blood cell production occurs in non-malignant states, or how this process might be impacting rare blood disorders. But over the past decade, thanks to a lot of work, including work from this community, particularly from Steve McCarroll, Ben Ebert, and others, we've learned about the fact that somatic mosaicism, we know, is widespread in all of our tissues as we age. And this has particularly been notable and well worked out in the context of the blood system, where there can be cancer driver mutations acquired in blood stem cells that then result in clonal expansion of the stem cells, even in the absence of an overt malignancy, a condition that's been termed clonal hematopoiesis of indeterminate potential. And approximately 10 years ago or so, when this condition was first described, it was thought to be an invariable precursor to the development of a variety of blood cancers, including myelodysplastic syndrome and acute myeloid leukemia. It was also known that this condition could predispose people to a variety of other comorbidities, including cardiovascular disease and other conditions. But this progression, of course, as shown here, was thought to just be an inevitable evolutionary process. And yet, over the past several years, we've learned that actually it's very hard to predict, even if one individual has such a mutation, what is the likelihood of going on to either see a clone with one of these drivers, such as DNMT3A, TET2, and so forth. Is that clone going to expand? And we know that even for individuals with the same exact driver mutation, there can be no expansion over time, regression of the clone in some instances, or there can be some growth at variable rates. Moreover, as shown in the y-axis here from this study in the UK, we know that individuals have variable rates of progression to blood cancers, such as acute myeloid leukemia, with some individuals having no increased risk, even if they have one of these driver mutations compared to the general population, and yet other individuals, even with the same driver mutation, having some increased rate. So what underlies this variation? Well, we know of a variety of factors. So we know from both observational and interventional studies in humans, as well as studies in model systems, that chronic inflammation and some infectious exposures can actually have a role in enabling some clones to expand in circumstances. And so certainly this has been and continues to be well studied and is really the subject of a variety of studies in model systems. We know that exposure to some types of chemicals and radiation can actually allow some types of clones to expand, particularly in the context of treatment with chemotherapy for solid tumors. We know that clones with mutations in TP53, PPM1D, and CHEK2 can often be seen to expand in those settings. But what about the role of germline genetic variation? Could there be either genetic variants within that are inherited that either affect the likelihood of a clone expanding or that protect an individual from that expansion? And so we thought perhaps we could try to identify such variants. And this was the work of a medical fellow in our laboratory, Gaurav Agrawal. And Gaurav got interested in this problem and really wanted to try to understand not just what were the variants that were there, because there had been other efforts to try to identify those, but really what were the variants that were most either protective or put individuals at the greatest risk of progression or clonal expansion with clonal hematopoiesis. And so Gaurav took studies that had been both previously done from a variety of biobanks, as well as other studies that we and other colleagues could do in a variety of large biobanks, and could, from a population of about 700,000 or more individuals, look at individuals with detectable clonal hematopoiesis across these various studies, and then just ask what variants were either putting individuals at risk of having clonal hematopoiesis or what were protecting individuals. And this really allowed him to look at a variety of variants, many of which were increasing the risk subtly but significantly for acquiring clonal hematopoiesis. But Gaurav got particularly interested in this variant on chromosome 17 that actually had had the most substantial protective effect against clonal hematopoiesis. Now, looking at this haplotype, this had a allele dose-dependent protection against clonal hematopoiesis, with individuals having 2 copies of this haplotype having about a 30% reduction in acquiring clonal hematopoiesis. This was protective in contrast to some of the other variants that have been described as protecting against specific types of clonal hematopoiesis. This was protective against all the major forms of clonal hematopoiesis we could see, including DNMT3A, TET2 mutant clonal hematopoiesis, ASXL1 and JAK2. And importantly, and in contrast to what had been seen for other variants, this protected individuals from acquiring a variety of blood cancers, including from acute myeloid leukemia and the myeloproliferative neoplasms, as well as all forms of myeloid malignancies as far as we could see from these large biobanks. In addition, there was a subtle but significant decrease in all blood counts, where we could see that there was a reduction in platelet counts, a variety of white blood cell counts, as well as red cell counts. But even in individuals with 2 copies of this haplotype, the counts still remained in the normal range that's typically seen in this setting. If we then looked across a variety of other phenotypes, other than the impact on clonal hematopoiesis, the impact on blood counts that I mentioned, as well as the protection from blood cancers, we did not see any other phenotypic associations with this particular variant, really suggesting that it was relatively selective for its effect in hematopoiesis. So how was this variant acting? And finally, I just mentioned one other thing, which is this variant was actually found across almost all global populations. But I would just note that this has been maintained at relatively low frequencies, with the highest frequencies seen in a number of European populations, typically in the 4 to 6% range, with other populations having this variant suggesting an ancient origin, but suggesting it was maintained at relatively low frequencies. So how is this variant acting? Well, if we looked within the haplotype, there was only a few variants within that haplotype, and only a single variant we found that was actually overlapping any kind of functional annotated region. And in this particular case, it was a region that was actually accessible within hematopoietic stem cells. This was also a region that was bound by GATA2 and a variety of cofactors that are known to be important for transcriptional regulation within blood stem cells. We could also see that this region of chromatin accessibility was selective for early blood stem cells and very primitive progenitors, but then this region became inaccessible as blood stem cells underwent further differentiation, really suggesting that this was selectively accessible within that compartment. And in addition, if we used either correlations between attack sequencing and RNA sequencing, or looked at long-range chromatin looping data, we could see that this regulatory region had interactions with the promoter of MSI2. Looking broadly within the region and over several megabases that surrounds this particular variant, what we could see was that MSI2 was the only gene expressed within blood stem cells, and none of the other genes in that local region were expressed. in this context. If we looked at correlations between the degree of chromatin accessibility in single cells in that region and the expression of MSI2, we saw a nice correlation between those, really suggesting that this was a regulatory element affecting MSI2. But to directly get at that, we could take blood stem cells from healthy human donors, introduce CRISPR-I constructs that allow us to transiently suppress gene expression from a regulatory element within these primary stem cells. And we can see across a variety of guide RNAs targeting this regulatory element that we could selectively suppress MSI2 expression, but we did not see changes in any other gene expression locally. Now, MSI2 has been really well studied by a number of labs, some of which I'm describing here, as being critical for progression in a variety of blood cancers and other cancers, and has been known to be important in some stem cell populations. But our goal is really to understand how is this variant acting to protect individuals from both acquiring clonal hematopoiesis and from going on to develop blood cancers. And so to start to get at that, we really wanted to try to delineate what are some of the variant effects. So at first, we took this regulatory element with both the reference and the non-reference allele. We could put it upstream of the endogenous MSI2 promoter, and then put this into blood stem cells from healthy human donors, and then actually examine what is the activity of this regulatory element within a context that's relevant, these primary hematopoietic stem and progenitor cells. And critically, what we saw was that this enhancer element did act as a typical enhancer in this exogenous assay, where we could see an increase in regulatory element activity. But this was partially suppressed by a twofold by simply introducing this variant associated with resilience in individuals, and that protects individuals from acquiring either clonal hematopoiesis or these blood cancers. When we looked at the variant effect, we could see that this actually disrupted a canonical GATA motif that is bound by GATA2 and is the typical motif for this. And indeed, if we took heterozygous donor stem cells, pulled down GATA2, we could actually see reduced occupancy of GATA2 in the allele associated with resilience from clonal hematopoiesis, as well as protection from blood cancers. And similarly, if we took accessible chromatin reads from individuals who are heterozygous for this variant, once again, we can see that there was a reduced chromatin accessibility at this element, really suggesting that this was disrupting a canonical GATA2 motif that was critical for binding of GATA2, and thereby resulted in reduced both enhancer activity as well as reduced GATA2 occupancy. Now, unfortunately, we did not have a direct way to precisely edit this particular variant in, but we could show, using our reporter systems, that introducing small microdeletions into this element actually mimicked the effect of the variant. So we could create these small microdeletions with paired guides within the enhancer, and we could also target MSI2 directly. And when we do this, we see that there's about a 20 to 30% reduction in MSI2 expression within the stem cell compartment at the mRNA level. And we can also see by intracellular staining for MSI2 protein that within this stem cell compartment, there's a reduction in protein levels, either subtly by the enhancer or more profoundly by directly targeting MSI2 itself. So what were the phenotypic consequences of this on the blood stem cells? Well, if we initially look and perturb either in this case adult or cord blood derived stem and progenitor cells from the blood system, we can see in this case in the context of adult stem cells that there's a reduction both in the fraction of cells that are these long-term stem cells, that this is using phenotypic markers, and we can see a more profound reduction by directly targeting MSI2, and this was also true if we look at the number of stem cells that's present. We could also see similar results, which I'm not showing you here, from cord blood, and so this occurred across a variety of stem cell sources. However, ultimately, our gold standard in the field of stem cell biology is to take these cells from healthy human donors or that are manipulated and transplant them into immunodeficient and KIT mutant mouse recipients and then wait 4 months for long-term engraftment. And so if we introduce an equivalent number of cells, either with the edit at a benign safe harbor locus, AAVS1, or in this setting with a microdeletion of an enhancer or targeting the knockout, we actually see that if we wait 4 months, there's a reduction in human engraftment in a dose-dependent manner depending on the degree of reduction of MSI2 as shown here. This was true across all lineages, so we had reduced engraftment across the stem and progenitor cell compartment as marked by CD34, the B cell compartment is marked by CD19, as well as the myeloid cell compartment is marked by CD33 as shown here. But interestingly, if we took the marrow of the primary donors and then actually replated them for colonies, we actually saw an expansion of the myeloid cell colonies. And importantly, we did see that there was actually a skew towards myeloid differentiation. And this was consistent with what we've known from studies in mouse systems, where it's been shown that MSI2 is critical for the balance between self-renewal of stem cells, producing more of themselves, and their ability to undergo differentiation towards the myeloid lineages. And so this resilience variant seems to actually be reducing the likelihood of a stem cell undergoing symmetric self-renewal and make it more likely that that stem cell will undergo differentiation. So what were the underlying mechanisms by which this was acting? And to start to get at that, we could conduct single-cell RNA sequencing in a sorted compartment of the stem and progenitor cells enriched for stem cells, and then we could examine differential gene expression within molecularly defined stem cells in that compartment. And this allowed us to find a number of genes that were differentially expressed, including most notably the most downregulated gene was reassuringly MSI2. And so this allowed us to look at gene expression changes by modeling variant effects in this setting in primary human hematopoietic stem and progenitor cells. But we also wanted to understand, since MSI2 was a well-known RNA binding protein, what were its targets in this setting? And so teaming up with Michael Karas and his lab at Memorial Sloan Kettering, we could use this approach called hypertribe, where we could hook MSI2 to an ADAR editing enzyme. And this allowed us to look at endogenous targets of MSI2 by turning this RNA binding assay into essentially a sequencing problem. And by deeply sequencing primary human hematopoietic stem and progenitor cells with this construct present, we could identify about 3,000 mRNAs or so that were bound by MSI2 or putatively bound by MSI2 based upon the edits. And critically, we could find many more targets that had been identified before, but these targets matched the known binding sites from other orthogonal approaches like ClipSeq and so forth that have identified the targets. And critically, these were enriched in the 3' UTR of these mRNAs as well. We found that by looking at the putatively edited targets, if we looked at differential gene expression, there was a downregulation on average of this gene expression. So this allowed us to really focus in on a group of genes that we could show was both downregulated upon perturbation of the MSI2 enhancer, but that was also edited upon introduction of this hypertribe construct. And this identified both known targets of MSI2 as well as some novel targets, including some key factors important for stem cell self-renewal and factors that were important for the regulation of stem cells. And so this, of course, made us wonder if we have this set of about 200 mRNAs that are downregulated and bound by MSI2, could we learn something about how they're playing a role potentially in enabling the activity of this particular context. And so we could examine ribosome profiling data that we performed within primary human hematopoietic stem and progenitor cells. This allowed us to show that this set of about 200 mRNAs was actually amongst the most highly translated mRNAs within the hematopoietic stem cells of human donors. But moreover, we could also show that this particular set of mRNAs, if one looks at mutant cells versus non-mutant cells, for example, in TEC2 clonal hematopoiesis, was actually upregulated. And we could see this across other drivers such as DNMT3A, ASXL1, and so forth. Moreover, if we looked at a variety of AML survival data akin to what's seen with MSI2 expression itself, this set of genes was also associated with worse survival with increased expression associated with that. But what about the cooperativity between this particular resilience variant and the acquired somatic mutations that occur in individuals? And so we teamed up with Alex Bick at Vanderbilt University and Yash Prasad in his lab. And as you can see from the data I showed initially from George Vasilio's lab, you can see that individuals with clonal hematopoiesis have a variety of annual growth rates. And here we're following individuals for a median of 6 years in the BioView cohort. But individuals with even one copy of this variant actually had decreased growth rates of these clones in the setting of having pre-existing clonal hematopoiesis at time 1 and then following them to the second time point. In addition, where individuals had detectable clonal hematopoiesis at time point 1 but then had disappearance of that clone, we found that this disappearance was much more likely, 1.8-fold more likely, in the setting of having this resilience variant. So not only is it protecting individuals from acquiring clonal hematopoiesis, but actually makes it more likely that that clone will disappear. So could we model this? Well, we could go on and edit ASXL1, just as a typical driver mutation. If we do that and we co-edit this benign safe harbor locus AAVS1, we see that the stem cells expand with this driver mutation. We could suppress that expansion in part by editing the enhancer that I'd shown you before, and almost completely suppress that by editing MSI2. We could see in this setting, moreover, that we could see colony replating if we introduce secondary colonies, really a feature of self-renewal that we're seeing, and this could be at least in part suppressed by editing the enhancer. And moreover, if we performed long-term engraftment of these cells into xenotransplant recipients and just waited, we could see an expansion of the primitive stem cell compartment, and this could be at least in part suppressed by editing the enhancer. Moreover, we could also use the reciprocal approach in collaboration with Michael Karas's lab, where we could take an ASXL1 mutant mouse recipient. Maybe I'm not pointing this in the right place. Maybe I can just go forward somehow. Doesn't seem to be. Well, it'll maybe let me go forward. But using this approach, we could actually, I didn't do anything bad. Oh, there we go. Seems to be doing something perfect. So we could see that thank you. So we could see that by introducing an ASXL1 mutant mouse and then increasing the expression of MSI2, that these mice actually have an expansion of the stem cell compartment, whereas ordinarily these mice do not have any phenotype. And at about 8 months, we see that there's a reduction in a variety of blood counts and a variety of signs of the fact that these mice then go on to, oh, perfect. These mice go on to have features consistent with myelodysplastic syndrome, including dyserythropoietic changes and Pelger-Hewitt type nuclei in the neutrophils. And so what I've shown you really is how by really deeply diving into a single variant, we can start to learn about mechanisms that might actually both protect individuals from acquiring somatic driver mutations in a condition that we know as clonal hematopoiesis, as well as that prevent progression of those clones even once acquired, as shown both based upon our longitudinal profiling data of individuals as well as what we could model. And I think what it really suggests for us is, of course, this is not the only protective variant, but certainly I think it suggests that by really deeply understanding this, maybe we could identify strategies that not only allow us to target cancers, but actually potentially prevent cancers in the future, as suggested in this diagram in this poster from 1932 from Paris. And so our hope is really by understanding some of these variant mechanisms, what occurs in individuals and what heritably allows individuals to be protected from cancers and pre-malignant conditions, that we could understand more about this process. And so with that, I just wanted to finish up by acknowledging the people who've really contributed to this work, particularly Gaurav Agarwal, a number of other lab members and our collaborators, as well as our funding sources. And I want to thank you all very much, and I'm happy to take any questions. Thank you.
Remarkable talk and grace under fire, so even better. Any questions for Vijay? Yeah, go ahead, Brad.
Yeah, that was awesome. So the variant, it's maybe just a random, unfortunate, or I guess in some cases fortunate event for individuals. Are there other selection pressures? Like, do you see this as just a random mutation? Do you to have a phenotype in other tissues?
Yeah.
If it's protective of leukemias and stuff, would it ever be selected for? Is there not enough,
Yeah. So, this is an inherited variant that's clearly occurred like early in history. It's interesting that it's been maintained at low allele frequencies. As far as we could tell, though, like I mentioned with the phenotype-wide association studies, there's no other major phenotypes that it's associated with. But we just have to remember that, we're all related. Probably, we have common relatives about 3,000 years ago. And, probably some of those pressures that our relatives had to go through, exposure to a variety of infectious agents, this might not have been a beneficial, variant in this setting. But in the modern era where we're worried, where we live, nice, hopefully long lives, and are relatively protected from some of those issues, hopefully this by mimicking this, especially if there's lots of drug developers, this is out of my, area of expertise, but certainly this could be a potential good target by mimicking this effect. And so I think that that's really our hope. Yeah. So Brad was asking, are there populations at altitude or other places? As far as we could tell, this has been maintained at relatively low frequencies across all populations, but is found across all populations.
Questions for Vijay? All right, well,
all right,
thank you, Vijay.
you.
Excellent talk, lovely biology. As promised, thanks for delivering.